Hi, my name is Nur Asma Atira Binti Paiming.
Hi, my name is Nur Amiris binti Faizi,
Hi, my name is Nuri Zaki Udaya binti Ayu.
and today we will be presenting forensic entomology.
Hello everyone, today I will begin this presentation by introducing the field of forensic entomology, which is the use of insect and other arthropods in criminal and legal investigation. So, forensic entomology is especially important in case of involving decomposed or mummified remains, because insects are often the first and most consistent biological evidence found on a body. When a person is dies, insects start to arrive in a very predictable sequence. This pattern allows forensic experts to use insect development and species identity to estimate the times since of death or the postmortem interval, also known as PMI. So, two major insect groups play important roles in entomology. The first group is Diptera or flies, which are usually the earliest insect to colonize a body. They can arrive within minutes or hours after death. The second group is Coleoptera or beetles, which typically arrive during the later stages of decomposition, especially when the body becomes dry or mummified. This makes beetles especially important in advanced stage investigation, like the case study we will be discussing today, which involves beetles found on mummified human corpse in Malaysia. So, overall, forensic entomology provides objective and scientific information that helps investigators estimate the time of death, determine whether a body has been moved, and better understand the circumstances surrounding a case.
Today, I will be presenting a unique forensic entomology case study published by UiTM researchers in the legal medicine in 2023. This case is especially valuable because it's documented for the first time in Malaysia, and two specific beetle species recovered from a human corpse. Species that had never been officially recorded on a humans remains in our country before. This case involved a mummified adult male found indoors in Selangor. The body showed signs of traumatic chest injury, but the exact time of death couldn't be estimated using a normal decomposition observation because mummification slows down or even freezes visible decay. That's why entomology became a crucial tool in these investigations. So, the environmental data was taken from the nearest weather station, and the average temperature was around 27.9 degrees Celsius with a high humidity. Because the body was located indoors, decomposition was slower, and this condition created an environment typically associated with the phorid flies and late stage of beetles, a pattern commonly seen in the concealed or indoor death scenes. So, during the autopsies, several insects were collected, and the earliest colonizer identified was Synthesiomia nudicenta, a music fly often found in tropical Indonesia. Its empty puparia were the oldest evidence the body and played an important role in calculating the timeline. The most abundant species was Megacelia species, a phorid or scuttlefly, and well known for entering small hidden spaces and appearing on mummified remains. The surprising and scientifically important finding was the presence of two beetle species, Dermates maculatus and Necrobia rufi pies. This discovery is significant because it marks the first confirmed base Malaysia where the both beetles were collected from human remains. Using developmental duration of Synthesiomia nudicenta at similar temperature, the research team calculated a minimum postmortem interval of around 16 to 17 days. This estimate was far more accurate than relying solely on the physical condition of the body. This case was not reported in the public news because forensic case details, victim identity, and autopsy information must remain confidential. Only anonymized scientific data is allowed to be published. Therefore, the full details exist only in scientific publication, which makes the thesis academically strong and ethically compliant. Overall, this case contributes a valuable entomological data to Malaysia and highlights the importance of insect succession in a forensic investigation. Now, I will explain the forensic importance of each insect species found in this case. This insect provide crucial clues about the decomposition stage, the environment, and the death timeline of death. The first species is Synthesiomia nudicenta, a music fly commonly associated with the indoor death scenes in tropical climates. This species does not usually appear immediately after death, but once it colonized, its development is predictable. The empty puparia found on the body showed that at least one full life cycle had been completed, making this the most reliable species for calculating the minimum postmortem interval. So next is Megacelia species or scuttleflies. These flies are extremely small and can enter through the tiny gaps, which is why they frequently appear in concealed bodies, a sealed room, or mummified conditions. Their presence supports the fact that the body was indoors and had reached a dry, advanced stage of decay because the thrive in the places that larger blowflies cannot access. Megacelia species are often dominant in indoor forensic cases. The third species is Dermastes maculatus, also known as the skin or hide beetle. This beetle feeds on dry tissues such as skin and tendons, making it a strong indicator of late-stage decomposition or mummification. Its serrated elytra and black and white ventral patterns make it easy to identify. Although the Dermastidae family is known worldwide, this is the first officially documented case of Dermastes maculatus recovered from the human remains in Malaysia. Lastly, we have Necrobia rufipis, the red-legged named beetle. This beetle has a metallic blue body with a distinctive red legs. It is predatory and feeds on maggots and a dried fly, so it typically appears only after the body has dried out significantly. The development data for this species is extremely important for the long-term PMI estimation, and this discovery supports the classification of the remains as a mummified. Together, these four species create a complete entomological picture: early Indo-colonization by music fly, continued activity by forage flies in the restricted spaces, and finally the arrival of late-stage beetles feeding on the dry tissues. This combination strongly supports the calculated MPMI and gives a scientifically confident reconstruction of the circumstances surrounding the death.
For sampling and preservation procedures, once collected, the insect were preserved following standard forensic entomology methods. Although the article did not list every step in detail, typical preservation procedure involve killings of bodies specimen like maggot with hot water to maintain their morphology, and then storing them in 70% ethanol. So, this prevents decay and preserve important identification features. Hard-boiled insects such as beetles are also stored in ethanol after collection. Each sample is labeled with important information such as the body region where it was found, its life stage, and the time and date of collection. In this case study, the human remains were found in a mummified state, which corresponds to the dry or skeletonization stage of decomposition. At this stage, most of the soft tissues have decomposed, leaving primarily dried remnants. The insect evidence collected support this stage. The beetles Necrobia rufipes and Dermastes maculatus are typical late colonizer that feed on dry tissues and are commonly found in mummified remains. On the other hand, the empty fly puparia indicate that Diptera colonized the body earlier, during the initial stage of decomposition. Together, this evidence tells us that body had passed through the fresh, blood, and active decay stage, reaching an advanced dry stage, which also aligns with the indoor tropical environment of Malaysia. In forensic terms, identifying this stage is important because it confirms that the postmortem interval was prolonged and explains why certain insect species were present at the scene. In this case study, the human remains were found in a mummified state, which corresponds to the dry or skeletonization stage of decomposition. At this stage, most of the soft tissues have decomposed, leaving primarily dried remnants. The insect evidence collected support this stage. The beetles Necrobia rufipes and Dermastes maculatus are typical late colonizer that fancy interpretation and PMI estimation. Now, let's take a look at how the postmortem interval or PMI was estimated in this case. PMI refers to the time that has elapsed since death, and insects provide key biological clues for this estimation. In this study, PMI was primarily estimated using fly development data. Specifically, the investigators collected empty puparia, and puparia represent the end of the larval stage, meaning these flies had already fed, matured, pupated, and emerged as adult before the body was discovered. By identifying the species and life stage present and applying known developmental timeline for this fly, the researcher could estimate a minimum PMI. In tropical indoor conditions, the time for flies to reach the pupal stage typically range from 7 to 14 days, with pupation taking an additional 4 to 10 days. Therefore, the presence of empty puparia indicate that colonization began to least 2 to 3 weeks prior, potential longer depending on indoor temperature and environmental condition. Beetles were also recovered in this case, specifically Necrobia rufipes and Dermastes maculatus. However, they were not used directly for PMI estimation because beetles usually colonize during later decomposition stage, such as mummification. Instead, their presence supported the finding that the body had been in an advanced stage of decomposition for a prolonged period. So, in summary, the combined evidence of fly puparia and late-stage beetles along with the mummified condition of their remains allowed the investigator to infer that the postmortem interval was at least several weeks, consistent with an advanced stage of decomposition.
So next, I will be presenting limitation and recommendation to improve these cases. So first, Malaysia has very few published forensic cases involving necrophagus beetles, so references are limited. Second, most developmental data we have come from overseas, which may not match our climate. And lastly, basic biological and ecological information for these species in Malaysia is still lacking. So to improve this area, the author recommend doing targeted research on the life cycles and behaviors of these beetles under Malaysian tropical conditions. And they also encourage publishing more case reports so we can build a stronger forensic references database for beetles in Malaysia also. So why this case is important? Because it's actually the first time that the Mastes, Megalites and Necrophobia rufipes were officially reported on human women in Malaysia. This confirms that these beetles do appear on local bodies, especially in late stage or mummified decomposition. So this gives us a new information about our own Malaysia forensic entomological. Next, this case adds to the forensic entomology knowledge because it shows that beetles also play a role in decomposition, not only flies. In this case, the beetles were found on a dry mummified body inside a house, which tell us how these species behave under tropical indoor condition. So this help expand our understanding of insect succession in Malaysia. And then lastly, this study highlight that we still don't have enough development data for these beetles under Malaysian temperatures. Without that information, it's actually hard to estimate PMI accurately. This case shows that we really need more local research to build proper growth charts and databases that match our tropical environment. So this is our last slide, which is for the conclusion. This case show that the Mastes, Megalites and Necrophobia rufipes are useful indicators of late-stage decomposition in Malaysia, but they're still not well studied. Their presence on a mummified body highlight their forensic value, but limited local data makes the MPMI estimation is very difficult. So more research and more case reports are needed to improve how beetles evidence is used in Malaysia forensic work. That's all from us. Thank you.